Welcome back to MLT 240. In today's lecture, we're going to be covering flagellates. We're still talking about organisms in the kingdom of protozoa. The flagellates fall in, of course, the kingdom of protozoa, but they also fall in the phylum Sauromastogophria. They fall in the subclass or subphylum of Mastogophria and in the subclass of Zoomastogophria. And in Zoomastogophria, these protozoa use a flagellate for locomotion. Here's a list of the flagellates that we're going to be covering in this lecture. Some are pathogenic and some are non-pathogenic. The ones that affect the GI tract are Giardia, Lamella, and Dientamoeba fragilis. The one that affects the genital tract is Trichomonas vaginalis, and it invades the vagina and the urethra. The non-pathogenic species that we'll be discussing is Pleomystis misneelii. First one that we're going to discuss is Giardia, and remember this one is going to affect a human's GI tract. This is the most common intestinal protozoa in the U.S. The method of infection is the ingestion of cyst in fecally contaminated food and water. This parasite has worldwide distribution, and it can survive up to 45 minutes in chlorinated water. And you can see what an adult Giardia looks like over here in this image on the right. So what are some clinical signs of Giardia? Sometimes people end up with what's called traveler's diarrhea as a result of Giardia. Symptoms can be anywhere from asymptomatic to severe diarrhea. Prolonged heavy infections can lead to malabsorption for a person. Stools will have heavy or large amounts of mucus usually. Symptoms can range from foul-smelling diarrhea. The people can also have foul-smelling gas, which is known as purple burps. It smells like rotten eggs. Also, some signs of traveler's diarrhea is abdominal pain, anorexia, nausea, vomiting, fatigue, and even weight loss from the severe diarrhea. In the lab, how do we ID a Giardia case? We want at least 3 stool specimens, which is pretty standard for any parasite analysis. You can also do permanent staining for each sample for a patient. You want to examine areas of mucus for the Giardia. Trophozoites adhere to intestinal mucosa via their sucking disc. So on the bottom side, they have like a little sucking disc or a little mouth that helps you ID the parasite. This usually results in intermediate shedding of the organism, so you may need 3 or greater specimens to be able to ID Giardia at times. There's also antigen-based immunoassays and fecal immunoassays available for testing. So what are these going to look like under the microscope as far as the Giardia cyst versus the Giardia troph? The cyst is going to be oval, and it's going to have anywhere from 2 to 4 nuclei, and they're usually going to be at one end. So here is a couple of the nuclei you can see in the cyst. The cytoplasm is retracted from the cyst wall. There's going to be a cluster of nuclei and a central axoneme. Remember, the axoneme is a core bundle of protein that helps make up the flagellum. So down the center here is the axoneme. That's the beginning of the flagellum that is ultimately going to form in the troph. And the cyst usually has an appearance of a little old lady with glasses or an old man with whiskers. And I don't know that these are great examples of that, but the cysts definitely look different than the trophs. As far as the trophs are more pear-shaped, and you can see that here versus the more oval cyst. They have bilateral symmetry. You have 2 anterior nuclei, and those are obvious right here. They have 8 flagella in 4 pairs. You can see all the flagella. They have a sucking disc on the ventral side, and they have fallen leaf motility. So under the microscope, if you see movement of the trophs of the Giardia, it looks like a leaf falling from the sky. It kind of twirls and moves slowly, just like a leaf falling. It has axonemes, so this line coming down the center that leads to the flagella, and the size is a little bit larger for the trophs versus the cyst at 12 to 15 microns. And these are usually easy to identify in the troph stage for Giardia. And you see the purple color of these guys? It's because these guys have been stained. That's why they appear purple in color. Up next is our next intestinal flagellate, Dientamoeba flagellis. This was first thought to be an amoeba, but it's actually a flagellate. And it was once thought to only produce trophs, but it actually has cyst forms as well. And you can see the life cycle down there in this image down below. Of course, it's going to enter through the mouth, going to go through the intestines, out, and the whole life cycle is going to start again. For Dientamoeba, it's usually asymptomatic, but patients can have diarrhea, anorexia, and abdominal pain. It also has an association with pinworm infections. The method of infection is unknown, but it's usually thought to be from hand to mouth or from fecal pathways. There is worldwide distribution of this parasite, and you can see over here what he looks like. A lot of times he looks kind of dirty or sometimes trashy looking, and that's kind of why his nickname is Mr. Trashy. And you can see a couple of nuclei there in the center. As far as the difference between trophs and cysts for Dientamoeba, the trophs have 2 nuclei that you can usually see, and is connected by a division spindle filament, and it appears to be filled with debris due to fragmented chromatin. Again, we refer to this one a lot of times as Mr. Trashy. It is due to that debris. The size is anywhere from 6 to 20 microns. The movement is sluggish and non-directional motility. No observable flagella, but it moves by means of a pseudopod or pseudopods. The cyst has organelles, including an axostyle. And remember, an axostyle is an elongated support rod that runs the length of flagellates. It has a flagella axoneme. Any flagellar components appear to be lost in excistation, which means the transition from cyst to troph, which allows the troph to move as an amoeba. The cysts are smaller, anywhere between 4 to 5 microns, and the precyst is up to 10 microns. There's usually 2 nuclei with large central karyosomes. Remember, karyosomes and endosomes can be used interchangeably, those terms, and karyosomes, endosomes mean a mass of chromatin in the nucleus. So if you look over here, don't let this image confuse you. These are the trophs. So the trophs goes with this information up here, and this is the little precyst stage, and then the cyst goes with this information down here. Our next parasite is Trichomonas vaginalis. It's the primary non-viral STI in the world. STI stands for sexually transmitted infection. It invades the female lower genital tract and the male urethra and prostate. Usually women exhibit symptoms and men serve as asymptomatic carriers. It can cause vaginitis and Urethritis vaginalis usually causes a prominent discharge. The incubation period is usually anywhere from 5 to 28 days. The transmission is usually by sexual intercourse, and distribution for trichomonas is worldwide. And you can see the image over here that the um, shows you the true form of Trichomonas vaginalis. For the lab ID, the sample is usually a urethral discharge or a vaginal smear, or you can even find Trichomonas in urine. The parasite is usually pear-shaped. There's usually a jerking motility or rippling motion, and this is usually caused by the undulating membrane, one of the defining characteristics of Trichomonas. Here is that undulating membrane. These trophs usually move pretty fast within a sample. There's no cyst form. Again, the trophs are pear-shaped. They have 4 flagella, 1 nucleus right here. You can see in the image. Right here it's the nucleus in these images. And you have this undulating membrane. In this slide, we're going to see the movement of the Trichomonas vaginalis. Again, you're going to be able to see that they move pretty quickly as a result of that undulating membrane. And you can see the flagella on the Trichomonas. And again, you can see these in a vaginal sample, urine, or a urethra sample. The final flagellate that we're going to talk about is Cleomastus misneeli. And as far as the cyst form, it has a lemon shape and it's got like a little bump on it that's referred to as the nipple. And that's this little bump right here that this arrow is pointing to. It has a single nucleus that you can see in each of these examples here in the image. It has a cytosome, which is the cell's mouth, and it's shaped like a shepherd's crook or hook. And that is what this is right here, and what this arrow is pointing to right here. That shepherd's crook or hook. And the troph form is more pear-shaped. The cytosome is more of a groove. It has a single nucleus with a central karyosome. It can have 4 anterior flagella, and it has a curved posterior end. So this end is more pointed, and this posterior end is more curved to make up that pear shape of the troph. Now we're done discussing flagellates. We need to move on and discuss the only ciliate that we're going to focus on for this lecture. And we are now in the class Kinetophragminifera. Again, we are still in the kingdom of Protozoa. We're in the phylum Ciliates at this point, but the species that we're going to focus on is Bacillium coli, so B. coli. And this species, or this particular class, the organisms move by means of cilia or little hairs. So let's look at some general characteristics of Bacillium coli or B. coli. It is the largest parasitic protozoan at 40 to 60 microns. It's the only ciliate that's parasitic to humans, so that's why we're only looking at this one ciliate. It can cause dysentery, which can be found, and it can be found in the feces, in the cyst, or the troph form, and is likely acquired through fecally contaminated food or water. It can also be a tissue invader. There's been reports of vaginal infections of this parasite. The diagnosis or ID is usually of trophs or cysts in feces or intestinal mucosa. It can multiply sexually by binary fusion or sexually in conjunction with exchange of micronuclei. And if you look over here to the right hand side, you can see two different images of the troph form of B. coli, and you can see all the little hairs on the outside that help with the movement of this parasite, the cilia that's on the outside, the little hairs. Now let's discuss the difference between the cyst and troph forms for B. coli. The cyst is usually not found as often, but there's a kidney-shaped nucleus that helps us identify the troph form, and that's shown right here in the image. And there's a micronucleus contained within this larger micronucleus that's kidney bean shaped. In the troph form, you still have that kidney bean shape of the macronucleus with the micronucleus inside. You also have the cilia, which is the small little hairs on the outside of the organism, which is the telltale sign you're looking at a troph. You have a cytostome, which is the mouth, which would be about right here on the organism, and you would have vacuoles within the organism. This concludes this section of the lecture for MLT 240. If you need clarification or need any extra help, don't hesitate to reach out, and I'll see you over in the next part.